Today we are going to have a class on reticulocyte count. What are reticulocytes? Reticulocytes are juvenile RBCs. Juvenile means that are young. Okay. Have you heard of that process called erythropoiesis? The formation of RBCs it starts from multipotent stem cells, and after formation of early normoblast, intermediate normoblast, then late normoblast, then comes this reticulocytes. Okay, reticulocytes are immature RBCs or juvenile RBCs. Okay, but here the nuclei are lost, but cytoplasmic RNA is present here. The cytoplasmic RNA is actually basophilic in nature. Basophilic, you know, basophilic means attracted to basic nature. Okay, so it is basophilic in nature, and as it is written here, see, reticulocytes are produced from late normoblast. Okay, but reticulocytes contain some basophilic cytoplasm remnants. Okay, but after the formation of mature erythrocytes, all of this gets extruded out, including the mitochondria, ribosomes. So this will not be seen in mature RBCs. This is only seen in immature RBCs called reticulocytes. Okay, short introduction. Now, what is the lifespan? What is the lifespan of a reticulocyte? Means for how many days would a normal reticulocyte be found in our blood? It is around 24 to 48 hours. Okay, or 1 to 2 days. In 1 to 2 days, it will mature into our mature RBC cells. So the count is around 0.5 to 1.5% of our total RBCs. In newborn, the count is a bit higher. It's around 2 to 6%. And after first year, this count actually starts falling, and it falls below 1% level, and that stays at that level till adult age. Okay, and we know, everyone knows now, the RBCs are formed from bone marrow. So reticulocytes are also formed from bone marrow. So the formation of reticulocytes actually indicates the function of bone marrow. If there are more reticulocytes in the RBC, in the peripheral smear, or in our blood, so what can you say? That the bone marrow is functioning normally. Okay. A little bit about the physiological significance. Of course, it is a physiology class, so we need to know about the physiological significance. Okay. So the number of reticulocytes, I have already told you, the number of reticulocytes count in the peripheral smear indicates the function of or activity of bone marrow. If it is very less, that can mean, actually there are more reasons for that, but one of the reason is that the bone marrow is not functioning properly. If the bone marrow is not functioning properly, the erythroid precursor cells cannot form. From that, the formation of these reticulocytes will be decreased. Okay, so for normal presence of the normal number of reticulocytes in the peripheral smear or blood, we need the adequate function of bone marrow. Okay, yeah, that is actually written here. And what is this reticulocyte response? Suppose you have anemia. Okay, and like for example, the most common anemia in our country is iron deficiency anemia, due to deficiency of iron in our diet. Okay, so you have iron deficiency anemia, you are given iron tablets, ferrous sulfate, ferrous ascorbate, or injection of iron. So now your bone marrow will act more. Okay, it has now iron. What was lacking before, now it has that. Okay, the iron is there now, so the bone marrow will now produce more amount of RBCs. So before formation of more amount of RBCs, the bone marrow will try to hasten up or speedily produce RBC. In that case, more amount of this reticulocytes will be seen in the peripheral smear or in the blood. So this is called a reticulocyte response. After therapy, see, I have diagnosed a case of anemia, I have given treatment. After treatment, there is a response. Okay, so in response, the reticulocyte count is increasing. After a therapy, reticulocyte count is increasing. This is called reticulocyte response. This is observed in case of pernicious anemia and iron deficiency anemia. I have already told you. So for practical purpose, now you are coming to the practical purpose, like what are you going to do in lab? You are not going to do that, but in lab what they normally do, now we will see what are the requirements for every practical. We first know what are the requirements of the practical. For every hematology practicals, we need that equipment for sterile finger puncture, microscope, watch glass, glass slides, and microscope to look. Okay, and here we come to know about a new term called supravital stains. Okay, stains means it is going to stain that blood. So then what is the meaning of supravital? Supravital means it is going to stain the living RBC cells, the living cells. How? This stain has a specific characteristics. It will enter into that cell and stain that cell. I have told you there are something called basophilic remnants of RNA in that reticulocytes. This stain is going to stain that basophilic RNA present in the reticulocytes. Okay, these are called supravital stains. Examples are new methylene blue, brilliant crescent blue. You can write it if you want. The procedure is here. It is written in short. At first the glass slides needs to be cleaned, and but here we are going to take that stain first, not the drop of blood. Okay, at first there is going to be stain, means that stain can be brilliant crescent blue or new methylene blue. We are going to take that supravital stain, put the stain on the slide. After that, make a sterile finger prick at the drop of blood to the stain and mix the blood with stain gently. Okay. Now cover it with a watch glass lined with moist filter paper for 15 minutes to prevent evaporation, so that it doesn't evaporate here. We have to keep it for around 15 minutes. But in practice, actually, what we do, we use EDTA given blood. EDTA mixed blood. EDTA is an anticoagulant. Okay, blood is taken from our venous blood or arterial blood, and then this reticulocyte is measured. So for that, to prevent coagulation of blood, we use something called anticoagulant. So anticoagulant mixed blood can be used. So first stain is given, then anticoagulant mixed blood is given, then it is placed for some time. Then we are making a smear out of it. After making a smear, we are looking through a microscope to find the adequate place to where we can see that smear perfectly. Okay, in some places of the smear there will see more amount of RBCs are there. In some places of the smear you will see the very less amount. We will see a place, we will find an area where there are adequate amount of RBCs. Okay, and they are not forming any Rowley formation. As in the previous class, ma'am has shown you na formation of Rowley, multiple RBCs stacked upon one another. So we will not see at that place. We will see where they are evenly spread. Now we will see through a wide immersion lens, as you already know about wide immersion lens in microscope under 100 power, and then we will see. And we will locate an area where there are approximately 100 to 150 red cells and see that in wide immersion field. Okay. Then we will try to identify the reticulocytes and RBCs. Now you can ask me how can you identify the reticulocytes from RBCs? Same thing. You will see in the next picture that RBCs are different from reticulocytes due to the presence of their basophilic cytoplasmic RNA. Okay. Then minimum of 10 fields will be taken and count a minimum of around 1000 cells. 1000 cells means containing both RBCs and reticulocytes. Okay. Then how are we going to calculate the retic count, the retic percentage? That is number of reticulocytes present in that smear field by the number of cells that you have counted into 100. At first we are going to take the stain. The stain we already know, brilliant crescent blue or new methylene blue. These are two supravital stains. Okay. I told you, supravital stains are that stains that are going to stain a live cell. It will go through the cell and stain what is inside that cell, the basophilic RNA content present in these cells. What are these cells called? RBCs and reticulocytes. Okay, so a drop of this stain is going to be put in a slide. Okay, so now after that we can use our own blood, or in practical purpose what they do, they take blood from our body, like they take blood by syringe and then they mix it with an anticoagulant like EDTA, sodium citrate, वगैरह वगैरह. They mix it with an anticoagulant so that the blood does not form clots. So this this EDTA mixed blood can be given here also. Okay, this blood is given with a drop. Now we are going to put it, cover that with a cover slip for around 15 minutes and cover that with a filter paper so that it does not evaporate. Okay. After that we are going to form a slide here. Smear, peripheral smear. I think 2 or 3 class later we will start this formation of peripheral smear, how peripheral smear is formed using 2 different glass slides. Okay, one glass slide there is drop of blood along with that stain, and along with using another glass slide we are going to form a peripheral smear. Okay, so that forms a uniform layer of cells. Okay, so what are you going to see here? What are you going to look under while immersion field? We are going to see around 100 to 150 red cells minimum. Okay, we are going to look for reticulocytes and RBCs here. Then we are going to calculate the retic count. Retic count is number of reticulocytes by number of RBCs into 100. And you have to take a minimum of 2 to 3 smears. One smear may not be enough. You may not be able to do that in first smear. So 2 or 3 smears needs to be made. Okay. So this is how they look under while immersion field. See, normal RBCs. See the difference between normal RBCs and that reticulocytes. Okay. See, reticulocytes have that, I told you, the basophilic staining of RNA content present in cytoplasm. They are visible here. Now, what I have already told you, the principle is here. The supravital staining used for reticulocyte count measurement, 2 stains, brilliant basal blue, new methylene blue. The RNA content of the reticulocytes can be detected by exposing the living cells to a supravital stain. What do they do? The living blood cells are mixed with stain and the stain enters the cell in living condition. Then the RNA present in the cytoplasm becomes precipitated and looks like this. Okay. And now we have come to a clinical part, like how can we find if it is hyperproliferative or hypoproliferative case by measuring that retic count. Okay. See, there is something called reticulocyte production index or RPI. Okay. RPI means production index. It is calculated by a formula that is written here, ARC by maturation correction. Now you may ask me what is ARC? ARC means absolute reticulocyte count. Now how are we going to calculate that absolute reticulocyte count? Please write it down. Absolute reticulocyte count equal to absolute reticulocyte count equal to retic percentage that we have just calculated. Retic percentage means reticulocyte count percentage into patient hematocrit by normal hematocrit. Hematocrit, HEMATO, hematocrit. You know, hematocrit is the total amount of RBCs in an entire blood. See, I am going to tell you again. ARC or absolute reticulocyte count equal to retic percentage into patient's hematocrit by the normal hematocrit or desired hematocrit. What is to be desired for that person in that age? Okay, this is the formula of ARC or absolute reticulocyte count. And from after calculating absolute reticulocyte count, we are going to calculate RPI, that is reticulocyte production index, or it is written as reticulocyte index. Okay, if the value comes more than 2.5, it is a case of hyperproliferation. See, that means the bone marrow are able to produce ample amount of reticulocytes, means bone marrow is working properly. Okay, if the value is more than 2.5, it means the bone marrow is working properly. If it is less than 2.0, that means the bone marrow is not able to produce enough amount of reticulocytes. Okay, so there may be some problem with our bone marrow. Got it? We are going to the next slide. So these are some conditions where the retic count decreases and some conditions where the retic count increases. This will be asked in the exam 100%. Okay. So what are the conditions in which the retic count decreases? What are the conditions in which retic count increases? Okay, I will just take 1 more minute. Please focus. So can you guess? Can you just say if there is any involvement of our bone marrow from where the cells are going to produce, new cells are produced. If that part of the bone marrow is damaged, or if that part of the bone marrow gets infected, or any malignancy, cancer affects that part of bone marrow, the production will be less. Okay, got it? The production will be less. So because of that, our retic count will decrease. Means our bone marrow cannot produce ample amount of reticulocytes or RBCs. That is written here. Bone marrow failure. Okay, like you see, have you heard of aplastic anemia? Aplastic anemia. Yes, aplastic anemia means when the bone marrow cannot produce any kind of cells, RBC, WBC, platelets. Okay, in that case, retic count is also going to be decreased. Malignancy. Malignancy means cancer. Folate deficiency. Folate means this is a vitamin B6 or sorry, vitamin B9. So folate is required for the maturation of RBCs. So if folate is not present, folic acid. Folate means folic acid. If folic acid is absent or folic acid is deficient, this will cause a decrease in the content of reticulocyte. Okay, everything has a reason. Okay, just don't memorize it. Just try to correlate why infections, why this malignancy, why folate, and why bone marrow failure causes this decrease in retic count. Okay, infections, infections involving that bone marrow will cause a decrease in the content of retic count. Now myxedema. Myxedema is something new for you. Myxedema means a case of severe hypothyroidism. This will also cause a decrease in retic count. And leukoerythroblastic crisis is similar to that of involvement of bone marrow by malignancy. What happens here? Leuko means WBC, erythro means RBC. So here both WBC and RBCs will be decreased. Okay, and in which cases the RBC, so reticulocyte count will increase? It's written here. In hemolysis, acute hemorrhage, pregnancy, and newborn. So two are physiological causes: pregnancy and young and newborn. Okay, but two are pathological causes here, like hemolysis and acute hemorrhage. Why do you think hemolysis will cause this increase in retic count? RBCs are continuously being broken down in our blood. So the demand for RBC is increasing. So something is breaking our RBC in our blood. That means RBC count is decreasing. So what will our bone marrow do? Bone marrow will say that we need more RBC. So they will try. Bone marrow will say, okay, I am going to make more RBC now. So before making more RBC, they will make more this reticulocyte now. So in that case, retic count will increase. That is our body response to some hemolysis. So you know about hemolytic anemia. Hemolysis causes anemia. Okay, hemolysis causes jaundice. So during hemolysis, our bone marrow will say, okay, let me produce more RBC. For producing more RBC, more reticulocytes needs to be produced, and that causes an increase in retic count. Similarly, during hemorrhage, suppose there is a condition in which your friend got into an accident, means road traffic accident. Okay, so he has lost lots of blood. So his entire RBC count is decreased. See, suppose there were so many RBCs in our blood, but lots of RBC got lost. So now he needs more RBC. So the bone marrow will try to form more RBC. So in that case, the retic count is going to be increased. Simple. Thank you.